Hi, welcome back to the FlowCytoscript tutorial. In this section, we're going to talk about cleaning up your data and exporting it in FlowJo for use in the script. So we're going to go over how to gate, label, and export your files in FlowJo so you can get the best results using FlowCytoscript. We're going to open our FCS files from our experiment in FlowJo, gate on the cells we want, and export just those cells into new FCS files. While there are ways of doing this directly in R, they're decidedly clunky. So why not just use software like FCS Express or FlowJo that are really purpose-built for this? We want to do this exporting process because it allows us to focus the analysis on cells that we're interested in, which makes the analysis more sensitive and much faster than if we include everything. At a minimum, we want to be excluding things like debris, doublets, and dead cells. You might wish to go further and select a specific cell type, such as exhausted CD8 T cells, and we'll go through a couple examples of that in this section. In the first example, I'm going to use these files I have here from a 51 color experiment using mouse cells run on the Aurora. So I'm just going to open that up here in FlowJo. I have, as you can see, 3 biological replicates, mouse 1, mouse 2, mouse 3, from 3 conditions: spleen, lymph node, and gut lamina propria. I've gated on the cells by forward scatter and side scatter, then single cells, first by side scatter, and then again by forward scatter. And then I've gated on viable CD45 positive cells. I've applied these gates to all the samples in the analysis up here. Now, if needed, I could create slightly different gates for different sample types, like the lymph node versus the gut, and by creating groups and applying samples, the gates to different groups. But I try to keep things as simple as possible and as much as consistently as possible. Once we've got these gates that we need to identify the cell type that we're interested in, what we need to do is right-click on the population that we want. In this case, these viable CD45 positive cells. So if I right-click, I get this pop-up menu. I'm going to select Export Concatenate, and we'll get this pop-up box. We're going to work under this Export tab up at the top, which will create one new data file for each of the original files in our workspace. Under the options, we're going to stick with FCS3 as output, but we're going to change the destination folder, creating a new one that with the folder name that's relevant for what we want. So I'm going to make a new folder here, which says FlowCytoscript 51 color tutorial. Okay. I'm going to say open. So now my destination is that folder. Now, next, under the parameters, we want to select all compensated parameters. We do want to use the compensated parameters, even if we're working with unmixed data. That's typically how they show up. The problem is that FlowJo doesn't actually always select this properly when we click this button. So I recommend using the option here, custom set of parameters, clicking on view edit, and then selecting select all compensated here. And that way you can actually see what it's selected. If you wanted to select different parameters, you could do that here. You can see down here, the uncompensated parameters are not selected for output. And we're going to say OK. Next, under the advanced options, we're going to click on that to expand the menu. Here we get the option to add tags to our files. This is really important because it allows us to add variable names to the files, which allows FlowCytoscript to know which group the files belong to. In my case, the groups are lymph node, spleen, and SILPL for the gut, which you can see are already encoded in those file names from the cytometer itself. So I'm going to delete this default export that gets prepended to the file names. You can see that the file name example that's being generated has export underscore at the top. If I get rid of that, I just get the file name with the population that I selected at the end. Now I'm going to click export. FlowJo should start creating new files in the folder that you put them in. And once this blue taskbar says complete, that we can close this window and cancel the other window. Okay, so that's our first example. We're now going to close this. For our second example, we're going to look at how to group files and add variable tags using FlowJo. So this one's going to be a subset of a human cohort analyzed using a 25 color symphony panel. So I'm just dragging in those samples here. If I expand this a bit, you can see that I have samples labeled P for patient and C for control. What we're going to do is actually add the tag patient and control to the samples. We could, in this case, actually select the files based off of P underscore and C underscore and separate them based off that. They do have identifiers written in them, but we're going to use this as an example of how to do it in case you actually didn't have that, or you wanted to put in identifiers that are easier to read. So what we're going to do is create two groups using this create group button or Control+G. The first group we're going to call patient. And we're going to create a group called control as well. If we wanted to sort our samples automatically into the groups, FlowJo has a lot of tools to do that here. We can select based off of keywords to have samples that go automatically into its group. You could change this to be the file name or the date that the files were required, or any other thing that has encoded into the FCS file that segregates the samples. And that would just populate the groups automatically with the samples. For now, we're just going to do it manually. So I'll create a patient group and a control group. At the moment, these don't have any samples in them, 0 samples. We're going to go to our all samples. We're going to put all of our patient files with the P into patient, and we're going to put those two control files from this small subset of the data into the control group. All right. Next, what we want to do is gate on the samples. And we're just going to do some quick gating to identify our T cells. So here we have our lymphocytes. We're going to do a scatter gate. And this is going to help us identify those single cells. And we're going to gate on viable cells so that we have our live dead marker. And we're going to go for CD3 positive, CD14 negative T cells. And as you can see, this data set is not perfect. So we're just going to do a quick gate like that. And that's going to identify our whole T cell population. Then we're going to apply this to each group. The gates will be identical, but we need to have the gates on the group for what we're going to do next. So now we've got our samples sorted into groups. We've got the gate that we want to identify the cells. We're just going to go through the exporting process again and add our tags. So export concatenate group. We're going to again select a new folder that is our patient cohort. We're going to select all compensated parameters. Say OK. And instead of export for this one, because it's the control group, I'm going to prepend control. And you can see now these samples will be labeled control. I'm going to export that. Those two are exported. Then I go to the other group and repeat the process, making sure to send them to the same location, select the same set of parameters. And here we're just going to call this patient. So these files will now be labeled patient, and the others will be labeled control. And if we go to our folder where I've just put those, we can see that we now have samples that are labeled patient and control that have been generated. So that's how you add variable identifiers to the file names. If you need to add multiple variables, for instance, you have patient and control samples from multiple time points or from multiple sources like blood and tumor, you'd want to add those tags sequentially, separated by an underscore. For instance, patient underscore PBMC and patient underscore tumor. That will allow FlowCytoscript to quickly grab a hold of those identifiers and sort your samples into the correct groups for you to do your analysis nice and easy. And that's all for this section. There's an optional section in the next step if you want to look at how to do transformation in FlowJo. Otherwise, the transformation will be applied automatically in FlowCytoscript, and really you can now just go straight to the FlowCytoscript workflow. And that's all.